Now we shall talk about certain important questions from immune system here now. The questions which were asked in the previous exams in NEET, AIMS, JIPMER, CMC, so on and so forth. Now here is the first question. I shall read out the question for you. CD4 receptors are associated with. The question is CD4 receptor is associated with A. AIDS, B. Cancer, C. Malaria, D. Pneumonia. Now the CD4 receptors, the T lymphocytes in human being are identified by means of two types of receptors. You call them CD4 receptors and CD8 receptors. CD stands for cluster differentiated molecules. Like T4 cells are identified by means of CD4, and cytotoxic T lymphocytic cells, which are also called killer T lymphocytic cells, they are characterized by the presence of CD8 markers. So here in an AIDS patient, AIDS virus mostly affects CD4 cells, that is helper T lymphocytic cells. Due to fall in helper T lymphocytic cells, B lymphocytic production also comes down. As a result, the person loses overall immunity and becomes vulnerable for various infections. So question number 1, correct answer is A. Now question number 2, assertion. Interferons are antiviral proteins. Reason, interferons are secreted by virus infected cells. Now in human body, T lymphocytes or any nucleated cell of the body, when they are invaded by virus, in response to the virus, they secrete certain proteins known as interferons. These interferons diffuse into surrounding cells and thereby make the cells resistant to those viruses. So they are widely used in medical field here now to cure rabies and different kinds of cancers, so on and so forth. So here both assertion and reason are correct, and reason is the correct explanation. So answer for the second question is B, that is both assertion and reason are correct, and reason is the correct explanation of assertion. Question number 3, I will read out the question for you. What is diapedesis? A kind of amoeboid movement. B, the process of filtration of urea in kidney. C, a type of locomotion found in hydra. D, migration of WBCs into the tissue spaces from blood capillaries. Now whenever there is inflammation or infection in the human body, the WBCs squeeze themselves and come out of blood capillaries to fight against foreign particles. So third question, correct answer is D, that is migration of WBCs into the tissues from the blood capillaries by squeezing themselves. So the third question, correct answer is D. Next you have question number 4. Cytokines that provide non-specific immunity against virus are A. interleukins, B. tumor necrosis, C. colony stimulating, D. interferons. Now the cytokines are certain proteins synthesized by the lymphocytes of the human body, and they command innate immunity. They command innate immunity or natural immunity of the human body. They provide resistance to the human body against various viruses. So among these the correct option is D, interferons. They are kind of interferons that are secreted by the cells of the human body, provide resistance to the human body against viruses. So the options here: A. interleukin, B. tumor necrosis, C. colony hybridization. C is colony hybridization or colony stimulation, it is given here. D is interferon. So the fourth question, correct answer is D. Now I will read out the question here now for you. Question number 5. The binding of antibodies to the antigen to produce large insoluble complex is known as A. antigen antibody complex, B. agglutination, C. immunization, D. suppressor cell reaction. Here when antibodies interact with the antigens now, preferably when the antigens are solid in nature, there will be formation of a solid complex, and that formation of a solid complex due to interaction of antigen and antibody is known as agglutination, because agglutination mostly results in formation of a solid complex which is insoluble. So, fifth question correct answer is B, agglutination. Now, question number 6 here now, you have an assertion here. Antigen can be easily recognized because it has antigenic determinant. Reason, recognition ability is innate. Every antigen that enter into human body or that are present in the human being have got certain antibody recognizing sites. Those antibody recognizing sites present on the antigen are called antigenic determinants, or they are also called epitopes. What are they also known as? Epitopes. So, these epitopes present on the antigen are easily recognized by the antibodies, and in response to the epitopes, antibodies have structures called paratopes. So, paratope or antigen binding fragment, it is called fab end. So, the fab end or paratope of antibody recognizes the epitope of the antigen. So, here the assertion is antigen can be easily recognized because it has an antigenic determinant. Reason, recognition ability is innate. That is, recognition ability is an inborn character, okay, present in human body. So, the moment the antigen enters human body, its antibody site present on the antigen, which is called epitope or antigen determinant, is recognized by an antibody or sometimes by other phagocytic cells. So, here the sixth question, both assertion reason are correct, and reason is the correct explanation of assertion here now. So, the answer for sixth question is B, that is both assertion reason are true and reason is the correct explanation of assertion. Now, question number 7 here now, assertion, cancer cells are virtually immortal because they have antigenic determinants. Reason, cancer is caused by damage to the genes regulating the cell division cycle. Now, cancer cells are immortal because they have antigenic determinants. Cancer cells here now, mostly they are caused by the viruses, or sometimes they may be caused because of some other inexplicable reasons here now. Okay. When the cancers are caused by viruses here now, okay, the viruses have epitopes, okay, and those viral epitopes can be easily recognized by the antibodies. So, they can be easily recognized and they can be killed by the antibodies here now. And the reason he says cancer is caused by damage to the genes regulating the cell division. So, reason is correct here now, but assertion reason, both of them cannot be correct answers. Cancer cells are virtually immortal. Cancer cells do not die. They escape from the apoptosis process, that is natural death process, because they are caused by certain viruses, and those viruses have antigenic determinants here now. Okay. And those antigenic determinants, even though they are recognized by the antibodies, they are not killed by the natural antibody which are present in the human body. That is why they keep on multiplying. So, here seventh question, assertion reason are correct, but their reason is not the correct explanation of assertion. So, the answer should be C, that is both assertion and reason are correct, but reason is not the correct explanation of assertion. So, seventh question option is C. Now, I will read out the eighth question for you. Antigen binding site in an antibody is found between A, 2 light chains, B, 2 heavy chains, C, 1 heavy chain and 1 light chain, D, either between 2 light chains or between 1 heavy chain and light chain depending upon the nature of the antigen. Now, when you study the structure of antibody here now, the structure of antibody was proposed by Rodney Porter. According to Rodney Porter, every antibody has a Y-shaped configuration like this, okay, where it is called, this is the heavy polypeptide chain, this part is called FC end, that is constant fragment or crystallizable fragment, and this part is called fab end, antigen binding fragment. This antigen binding fragment, apart from the heavy chain, this is the heavy polypeptide chain, heavy polypeptide chain, And here in the Fab end region, it has got even a light chain also. This is the light polypeptide chain. Light polypeptide chain. So heavy chain and light chain, both of them constitute Fab end, that is antigen binding fragment, which is also called paratope. So paratope or Fab end is a part of antibody to which the antigen gets stuck. So as per the question, antigen binding site in antibody is found between, so C should be the correct option, one heavy chain and one light chain. This is the heavy chain, the other one is the light chain. Both of them collectively form Fab end, antigen binding fragment or paratope to which the antigen is stuck. So here, eighth question, correct answer is C. Ninth question, I shall read out the question for you. The treatment of snake bite by antivenom is an example of A. Artificial acquired active immunity, B. Artificial acquired passive immunity, C. Natural acquired passive immunity, D. Specific natural immunity. Now the immunity in human being is divided into two types. One is natural acquired immunity, one is natural immunity or innate immunity, which is there in the child right from the birth, which is mostly inherited from the parents. The natural immunity or innate immunity, which is also known as, it is because of the phagocytic cells present in the human body or cytokines present in the human body, or maybe because of the skin or because of the mucous lining of various organs. So all those come under natural immunity in the human body. The other one is acquired immunity, which is also called adaptive immunity, which is specific in nature. This is acquired in human body maybe because of the antigenic invasion, or sometimes it may be because of prior contact with the antigen. So again it is divided into two types, active acquired immunity, passive acquired immunity. Under active acquired immunity you have natural active acquired immunity, artificial active acquired immunity. Natural active acquired immunity is immunity that builds up in the human body due to prior contact with the pathogen. Suppose a person who has suffered from smallpox or chickenpox develops immunity throughout his or her lifespan due to formation of antibodies. So when the smallpox virus or the chickenpox virus enters into the human body, it induces formation of antibodies, B lymphocytes, which are responsible for the formation of plasma cells and memory cells. Once sufficient number of memory cells are synthesized, as and when the same pathogen enters human body, that pathogen is killed outright by the memory cells which have been synthesized. So that kind of immunity is called natural active acquired immunity. That is immunity that crops up in the human body due to prior contact with the pathogen. Artificial active acquired immunity is immunity that builds up in the child due to vaccination. When the vaccines are given, those vaccines induce the formation of memory cells. Those memory cells protect the child against invasion of the same pathogen. So that is called artificial active acquired immunity. Passive acquired immunity, here the antibodies are formed in one host, and from that particular host they are transferred to the human being. That is, they are transferred from sensitized to non-sensitized host, from one sensitized host to another non-sensitized host. Again, these are two types, natural passive acquired immunity, artificial passive acquired immunity. In natural passive acquired immunity, there is a transfer of antibodies from mother to fetus through placenta during gestation period, or soon after delivery there will be transfer of antibodies from mother's blood to, or from mother's milk to the infant through milk. So during gestation, certain antibodies like IgM antibodies, IgG antibodies are transferred from mother's blood to the infant, that is the fetus, through placenta. Then soon after birth, as the mother starts feeding the baby, through milk also there will be transfer of antibodies like IgA antibodies are transferred from mother's milk to the infant through milk. So that comes under natural passive acquired immunity. Then artificial passive acquired immunity, the antibodies that are synthesized in one host, sensitized host, they are transferred to non-sensitized host. So under this, polyvalent antivenom comes here now. Polyvalent antivenom is synthesized in horse or sometimes mice by injecting small doses of venom of cobra, krait, Russell's viper and saw-scaled viper. So, the polyvalent antivenom given to the human being is a kind of artificial passive acquired immunity. So, the answer among these options is the correct option is B, that is artificial passive acquired immunity. So, now the question correct answer is B, that is artificial passive acquired immunity. Question number 10, here is assertion, WBCs accumulate at the site of wound by diapedesis. Reason, it is a squeezing of leukocytes from endothelium. So, whenever there is inflammation or infection in the human body, the WBCs squeeze themselves and come out of the blood capillaries to fight against the foreign particles. The process of expulsion of WBCs from the blood capillaries by squeezing themselves is called diapedesis. So, assertion is correct. WBCs accumulate at the site of wound by diapedesis. Reason, it is a squeezing of leukocytes from the endothelium. He is giving the meaning of the diapedesis, but the reason is not given here now. Reason, what should be the reason here now? Why do WBCs come out of the blood vessels? They come out of the blood vessels to fight against foreign particles. That should have been the reason, but reason is given different here now. But the question, both assertion reason are correct. That is, reason is it is a squeezing of leukocytes from the endothelium of the blood vessels to fight against the foreign particles. So, here 10th question, both assertion reason are correct, but reason is not the correct explanation of assertion. So, 10th question correct answer is B, that is both assertion reason are correct, but reason is not the correct explanation of assertion. Now, question number 11, when the word AIDS Day is celebrated, A. 7 July, B. 14 July, C. 1st December, D. 23rd May. 1st December every year is celebrated as World AIDS Day. So, answer is C. Now, question number 12, antibodies are produced by A. leukocytes, B. lymphocytes, C. erythrocytes, D. none of these. Mostly lymphocytes, B lymphocytes synthesize antibodies, bone marrow induced lymphocytes synthesize antibodies. So, 12th question answer is B. Now, 13th question, the most abundant immunoglobulins in our body are A. IgA antibodies, B. IgD antibodies, C. IgG antibodies, D. IgM antibodies. The most abundant antibodies found in human blood are IgG antibodies. They are found in blood, okay, and they are even transferred from fetus to from mother to fetus through blood. So, 13th question answer is C. Now, question number 14, I will read out the question for you. Formation of antibodies within our body is called A. passive immunity, B. active immunity, C. innate immunity, D. acquired immunity. Now, formation of antibodies is known as active immunity. Under acquired immunity, just now I have explained about the types of immunity in detail. Under acquired immunity, again there are two types now, active immunity, passive immunity. Under active, there is a natural active acquired immunity, artificial active acquired immunity. Natural active acquired immunity is the formation of antibodies due to prior contact with the pathogen, and artificial active acquired immunity is formation of antibodies by vaccination, formation of antibodies due to administration of vaccines. So, among these the correct option is B, active immunity, that is active acquired immunity. Now, question number 15, AIDS is characterized by a sharp reduction is A. helper T lymphocytic cells, B. killer T lymphocytic cells, C. suppressor T lymphocytic cells, D. B lymphocytes. AIDS virus, when it enters, that is a retrovirus, when it enters human body, it mostly destroys helper T lymphocytic cells which are carrying CD4 markers. Due to fall in helper T lymphocytic cells, B lymphocytic production also comes down. So, the main target for the AIDS virus is helper T lymphocytic cells. So, 15th question answer is A, helper T lymphocytic cells. Now, 16th question, which of the following fights against infection? A. RBC, B. blood platelets, C. WBCs, D. None of these. Mostly WBCs fight against infection by synthesizing antibodies, and some of the WBCs are also phagocytic in function, like neutrophils are phagocytic, then monocytes are scavengers. Okay, so 16th question correct answer is C. We have 17th question. Interferon is A. Antibacterial protein, B. Antiviral protein, C. Antifungal protein, D. Anti-snake venom. So the answer is B. Antiviral protein. Interferons are the proteins synthesized by cells due to infection by viruses. So they are antiviral proteins. So 17th question answer is B. Now question number 18. Which one of the following is allergic disease? A. Hay fever, B. Skin fever, C. goiter, D. enteric fever. Hay fever, seasonal allergic rhinitis it is also called. It is due to allergic reactions, Okay, so 18th question answer is hay fever. That is allergic disease. So 18th question answer is A. Hay fever. 19th question, I'll read out the question for you. Histamine is secreted by A. mast cells, B. fibroblasts, C. chromatophores, D. macrophages. Now the mast cells at the time of inflammation here now, they get ruptured to release various physiological mediators like histamine, heparin, serotonin, bradykinin, so on and so forth. So histamine, which is a vasodilator, is secreted by mast cells. So 19th question answer is A. Question number 20. I'll read out the question for you. Active immunity means A. resistance developed after disease, B. increased quantity of blood, C. resistance developed before disease, D. increasing rate of heartbeat. Active immunity is mostly due to the invasion of the pathogens or because of vaccination. So resistance developed after disease, that is a kind of natural active acquired immunity. So among these options now, the correct answer would be A. resistance developed after disease, which is also known as natural active acquired immunity. So 20th question correct answer is A.